Hello, everybody. This is Matteo di Nardo from Rome speaking. Good morning, good afternoon, good evening. Very happy to be here and present this important webinar together with my friend and colleagues Aparna, Hoskote, Roeleveld, and our great friend Alain. Today, we are going to speak of an important project related to the definition of pediatric adverse events on ECMO. And I'm going to introduce Ryan Barbaro and Peta Alexander, very well-known physician and friends. Ryan from the PQ of Arbor, the Mod Children's Hospital, and Peta Alexander from the Boston Children's Hospital. I'm gonna introduce again my co-chairs so that they can address something, and then we will start immediately the webinar.
Matteo, should we get...
Aparna, yeah, if you want to say something, if not, we can start.
Let's go ahead.
All right. Thank you so much, Matteo, for introducing all of us so kindly and generously. Peta and I are really excited to talk with you today about a project that we had the opportunity to think about just a short time ago. But the name of it is the Academic Research Consortium, ECMO Central. And the goal here, as you can see in the subtitle, ECMO Core Elements for Trials, Regulation and Quality of Life, is to help us try and, with your input and advice, create definitions, adverse event definitions for clinical trials. And so before we get started, Peta and I would just like to disclose that as we think about trying to craft adverse event definitions for clinical trials, that we do have roles both within ELSO and other research networks, PDECMO, and ongoing clinical trials that could inform this. And so we acknowledge those as disclosures as well. In terms of our agenda for the day, what we're hoping to accomplish here with you, I just want to give a brief overview before we move in. So our goal here is to give a little bit of background, which is what we'll jump into next, and then pause for some questions. And then we'll walk through 4 of what end up being several different adverse event definitions that we have as exemplars. And then we'll also share with you how we can find these beyond this, so that if you're interested in gaining more information, that you're able to do so. But in between, at each point, hopefully we'll have some good opportunity for questions. Our objective here is really to be able to develop harmonized adverse event definitions, as we said, for use in clinical trials and regulatory evaluation. And we think this is really important because if there are not harmonized definitions across clinical trials, and when those clinical trials are done, it's going to be much more difficult to assimilate the information across those trials so that they're able to be aggregated and hopefully improve on patient outcomes. And our scope really here is adverse events assessed to be associated with extracorporeal life support. And then our guiding principles as we try to move through this is intentional harmonization with existing definitions, modified where necessary to meet the needs of children. And so those existing definitions we'll talk to in a moment on some of the next slides. I'll ask Peta to do that. And then we'll here talk about the other kind of guiding principles are reproducibility, measurability, and efficiency. And if possible, we're going to try and avoid treatments wherever able in the adverse event.
And so as we began this process, Ryan and I actually learned a lot about a group called the Academic Research Consortium, and some of their previous definitions of adverse events for clinical trials are presented on this slide. Notably, an earlier definition of bleeding for cardiovascular clinical trials. These were all in adult populations and mainly adult interventional cardiology spaces. But you can see the red highlighted consensus statement is actually a set of standard adverse event definitions derived from mechanical circulatory support. And that paper, led by Bob Kormos and a group mainly centered in Intermax, actually proposed adverse event definitions for clinical trials of mechanical circulatory support for both adults and pediatrics. But at the point we became aware of the process, a pediatric MCS group was putting forward a new process for definitions specific to the pediatric MCS population. And Ryan and I were invited by the FDA to co-lead that process. As we recognized that they were keen to make adverse event definitions across pediatric mechanical circulatory support, mainly VADS, and ECMO, we were very keen to get an ECMO process together that would better represent the ECMO community, rather than have Ryan and I as the only engaged ECMO people in that group. And that's how ECMO Central actually was launched. The other existing definitions which are in use in clinical trials include the neurological endpoints for cardiovascular clinical trials and the third iteration of some valve definitions. But the bleeding definition has actually been used in many of the ECMO clinical trials that are already underway. So as we move forward with our process, we were intentionally planning to harmonize to existing ARC mechanical circulatory support definitions, unless there was a pediatric or an ECMO specific reason to divert. And where there was no ARC existing definition, then we were planning to reference existing ELSO definitions from the registry data definitions, and to consider whether there's a substantially better definition in the medical literature. One of the classic examples of that is for acute kidney injury, as you'll see with Dr. Gorga presenting later. The KDIGO definitions are so well established across that space, it seemed like we needed to integrate that sort of definition into anything that we brought forwards. Where a definition doesn't exist at all, we've really taken the opportunity to leverage the experts in our panel to start to build out new definitions. Next slide when you're ready, Ryan.
Sorry, Peter.
No, that's totally fine. Thank you for holding that. Would you like to talk to the methodology?
Sure. So in terms of how we were thinking about this, we wanted to make sure that we really talked to a bunch of people to help to inform just what we were doing, many of which are helping to lead this conversation today with us. So thank you. And so we talked to the ARC board and previous chairs just about what are their principles of approach to doing this process, because as Peta had mentioned, the ARC process to us a little bit unfamiliar. We talked to statisticians for of analysis, trialists to really try and identify those utilized definitions. And Dr. Combs, among others, had pointed us to the fact that the ARC definitions had already been incorporated in many of the adult clinical trials in the ECMO space. Librarians for consensus scoping review of the literature, and experts in consensus Delphi processes, of which Peta is, but also Labembia and Carol Hodgson and others, implementation scientists as well, because we wanted to make sure that what we created was both appropriate and feasible to this space. And we really tried to connect with quite a number of stakeholders as well. And we're quite excited about the willingness of people to sit and talk with us about this. We have engaged regulators from the FDA at the US, Canada Health, and the PDMA from Japan, as well as device developers, leadership from ELSO and ECMONet and PDECMO, funders within the NIH in the US and NIHR at the UK, biomedical engineers and translational scientists, families and patients, and a host of healthcare providers that have really been instrumental in helping us think about this as well. Peta, would you just maybe walk us through the topic groups that, yeah.
Absolutely. So we basically leveraged the existing MCS ARC definition manuscript to identify which topics we were going to assign experts to help to get together the existing definitions in the literature. And they're basically the topics that we chose are listed here. Bleeding is obviously hugely important for children on ECMO. Neurologic events was a key definition that we identified early. Acute kidney injury, device malfunction, hemolysis, infection, and vascular access related injuries are all identified now as draft definitions formed by our expert groups and discussed with our expert panel. We also addressed some other definitions, including non-CNS thrombosis, both arterial and venous thrombosis and thromboembolisms. Hepatic dysfunction was really challenging for us to look at on ECMO, and so that's still under development. We've developed a new definition of right heart failure on ECMO, as well as left atrial hypertension, lactic acidemia in the context of what we consider inadequate ECMO support, and similarly a hypoxemia definition in what we consider inadequate ECMO support. And so they're the draft definitions that we've got underway. And Ryan's going to go through our timeline of how we've landed to where we are today.
Yeah, so it's fitting that I kind of jumped the gun on you finishing that slide to get here, because we've been really just pushing hard ahead with this. The primary reason has been because there was this ongoing pediatric MCS ARC process that we wanted to harmonize with, and you'll kind of see these blue boxes down here about harmonization. And so what we've walked you through already is just the opportunities that we've had to reach out to leadership and methodology and measurement experts, as well as talk with stakeholders before we launched into a discussion of adverse events, which we just have finished up. And now kind of across this week of March, we are going before the community. We just talked within a US-based, or sorry, an international research-based consortium at ECMO, which has had a meeting in the US. We're really grateful for the time and the opportunity to talk with this with our European colleagues as well through EuroELSO. But our goal is to kind of globally gain good feedback on how we can work these processes and these definitions better, so that once we do obtain adverse event definitions that we're going to send out for voting, that we have something that really does meet the needs of the field and has been able to be grappled with and hopefully address some of the outstanding concerns as best we're able to. But after each of those, we will come back together as a leadership group and reformulate those definitions so that we can make sure that they are responsive and then try and harmonize alongside of the PEDS MCS process. So thank you everyone for the tremendous dedication of time and effort to the four who are here with us today, Dr. Gorga, Dr. Sporeto, Dr. Willems, and Dr. Rojas Pena, who will be joining us as well in just a bit to talk about the various different definitions that we have. So without maybe further ado, this is just a slide from our one of our early adverse event definitions. You can see we had a blast talking about definitions here, but no, we've really been tremendously excited and encouraged by the engagement of the community to help us to do this, and we really are hopeful that we can lean on all of you here today to continue that process of really engaging the community to move forward with the work. So Steve, if you're ready, I'll pull down my slides and have you share with us the definitions that you helped to lead the crafting of.
Just before Steve does, I think that we had actually allocated ourselves just a little bit of time in case there were specific questions about what the process was.
Thank you.
At this stage, I'm sorry. I think that one of the key things that we do want people to be aware of as we start the discussion is that what we're aiming to define here is adverse events for clinical trials. And if the definitions are acceptable and functional in a way that might work for registries, then obviously they may be incorporated into important registries in the space as well. But today, what we're really aiming to talk about is definitions of clinical trial adverse events. And that's particularly important at this time and in this space for pediatric ECMO, because there are actually a number of, at least one large and a couple of smaller clinical trials underway. And to be able to share information meaningfully across trials as more become available, it makes a big difference if we're all talking the same language with similar definitions.
Hi, thank you for including me. Maybe just a question on the stakeholders you mentioned. You mentioned that from the regulatory side, you included US, Canada, and Japan. Were there any specific obstacles or reasons why none of the European Union regulations were involved in this process?
So I think, to be fair, we actually tried everything we could to reach out to regulators across the world. We also reached out to TGA in Australia. We had some contacts, but no real communication with UK or European regulators. But we'd be very happy to hear from them if there's anyone, or if anyone has particular contacts because we are very much engaged in having their voice at the table and any input that they might have.
And just like, so, you know, that would really be meaningful right now too, because our plan is to share the draft definitions that we're presenting today with all of our regulators so that they can comment on them. And so if you were able to make that connection from somebody who is willing to kind of embark on this with us, they would still be able to have quite a formative process in giving feedback. Thanks for that question.
It's fantastic.
Just one more question for you all. With all these adverse events, and I'm thinking of, like, you know, most of our, you know, cardiac ECMO patients, have you consulted cardiology, cardiac surgery, in terms of, you know, how they would contribute towards that decision about definitions?
Yes, Aparna, even on our leadership group, we were really careful to include a mix of different subspecialties that could meaningfully inform the process. So in addition to Ryan and I and Matteo, we have Pedro Del Nido, who's a device developing cardiac surgeon with me here in Boston. We've got Angie Lords, who is the chief of the Action Heart Failure Network. And she's a cardiologist, and we have Adam Vogel, who's a general pediatric surgeon. And our experts across the world actually also represent all of the different craft groups that you might expect might be important for this project. And so I'm going to miss people if we go through a list, but in addition to neonatologists, pediatric ICU, cardiac ICU, cardiologists, all of the appropriate surgeons. We also have neurologists, neuro ICU specific people, radiologists, infectious diseases people, specific cardiologists who are both interventional as well as right heart failure, pulmonary hypertension specific. We have families and bedside nurses, perfusion, ECMO specialists. We've tried really hard to have as broad a representation as possible across craft groups as well as across the world. And I think that we have better achieved that sort of diversity of representation in this project than anything else I've been involved with in the past.
And Peta, if I could just build on that, I think, you know, some of the, so like, we wanted to make sure that anybody that we thought was going to be at the bedside caring for a patient, that we had involvement of those people, both across experience levels, so from professorial to, you know, actually trainees as well. And also we wanted to make sure that we had an international exposure across those bedside folks. We also, though, what Peta was talking about when we think about infectious diseases or radiology or, you know, hematology, we wanted to make sure that we also had folks that would really neurology, neurocritical care, help us to craft the definitions in the specific area. Actually, Steve has a group of folks who really are just world leaders in nephrology and neurocritical care. And the interplay between nephrology and critical care, both in adult and pediatrics perspectives, because sometimes the person who is the most equipped to help us think about the definition might not be a pediatrician. And so we really, and I think the other thing, we really want to say thank you a lot to Dr. Combs, who's joining us today. He spent some time with us early on to help us to think about how we, as we're crafting pediatric specific definitions, that we do that in a way that provides us an opportunity, hopefully, to be linked up well with what hopefully the adults are able to take forward from there, and connected us with many of the adult trialists to help us think about that. I don't know if you have any words to add there. I see you coming on, Alain.
No, okay, thank you again for the invitation. I think the project is a great one. We need this type of definition to move forward, to have research conducted on every continent using the same indicators, especially regarding side effects of ECMO, which remains a technology which confers some risks to the patient. And obviously from the scientific point of view, but also from the regulation point of view, we need all these data, and we need to have a common definition. No discussion about that. So we are strong supporters. I congratulate you again for conducting this initiative in pediatric patients, but we also will have at some point to move the same way for adults. This is a major point here.
But yeah, Aparna, thank you for your question.
Yeah.
And I'll look forward to that comment.
Just one more thing was, about involving secondary care providers, community, parent, representatives. That would be good to know if they were involved as well.
Absolutely, Aparna. That's been some of the most fun discussions that Ryan and I have had. We brought in mainly parents of patients who received ECMO. Ryan actually has a patient who received ECMO who's also a part of the process, and Malabembea actually introduced us to a current medical student who's an early survivor of ECMO in infancy, and being able to talk to her about the process that we're undertaking and the importance of it was really special. And so we have actually got those community people, parents and patients all involved, and some of them did actually join some of our adverse event discussions, but I think that where we've really benefited from their involvement was in some broad early discussions before we undertook the adverse event definition discussions themselves, to really help us have a high level view of what was important to them. They'll also look at the definitions again afterwards. And Ryan, I don't know if you want to speak to the recommendations that some of the parents gave us early on about what they'd like to see us do with this.
Yeah, so I think, one of the questions, obviously everybody comes to this from a different vantage point. The medical providers, you had also asked about ancillary health folks who we definitely have and we can talk about, but they have a different lens, and our parents do as well. And actually many of our parents have very intentionally different lenses. So Peta had worked with somebody who actually their child was on ECMO, but they're also an adult critical care intensivist. And then we also had somebody who's a nurse whose child was on ECMO as well. And so have some of that dual perspective, but others who really don't have a perspective. So one of the folks that we cared for was actually a patient whose child died. And we asked them, what is important to you in being involved in this? And they said, for me, I'm an advocate for my child. And what was really important and meaningful in me and being a part of this care team, which I felt a part of, was that I was able to advocate for what I thought would be best for my child. And leaving this, what I hope is that as my role in this is that I help other families to advocate for the care of their child. And so how does adverse events and advocacy line up? And I think what he and we had spoken to is that making the information that comes out of this accessible to parents and families so that adverse events that are important and meaningful to us as clinicians, obviously here through the lens of clinical trials, are things that parents can think about and be educated around and be able to speak to their care providers about, so that they feel a part of that conversation, a part of that work. I think another space and place where we asked and received willingness was, so we won't talk about this one today, but one of the places is neurologic adverse events. And so I think it's important to know whether somebody had a neurologic adverse event, but I think we all see an enormous spectrum on the impact of that adverse event. And we were talking with one of our neurocritical care doctors just yesterday, actually, about how we characterize that. And we were like, well, let's just go back to our families and ask them what's meaningful to them in terms of the impact coming out of this. So we are using their perspective to help make sure we think about this where it is, maybe that the adverse event has a patient-centered impact as well. Building on the second question, the ancillary health, I think the biggest space and place where we wanted to make sure that we're careful on that is those folks who are at the bedside at helping to provide the care, but come from maybe different perspectives. So perfusionists, respiratory therapists, nurses are all a part of that team, but also other members of the team who are certainly a part of our rounding teams a lot of times, or at least their day-to-day, social workers, pharmacists, psychologists, and those as well. So there may be more breadth or depth to that, but there's a good chance we've asked and just not mentioned, as Peta said, the entire list.
Thank you for that.
Yeah, thank you. I think it's great opportunities to reflect on something that was really important to us to make sure that we hopefully get a pretty holistic view of this.
Fantastic. Shall we move on with the presentation of the
So
bleeding?
what I anticipated 10 minutes ago will now do. Thanks. I appreciate that.
Great
Steve.
discussion.
Ryan,
Yeah.
do you want to just,
Yeah.
Ryan, are you happy to introduce
Of
Dr.
course,
Gorga?
absolutely. Yeah. Dr. Gorga is a colleague of mine at the University of Michigan and a tremendously thoughtful scientist, especially as it relates to the intersection of critical care and nephrology or acute kidney injury in particular. We asked him to come along and help us to think about this space. And he was really gracious and willing to do so. So thank you so much. And has brought together a tremendous team with incredible engagement and helping to craft a definition that I'll say, at least speak for Peta and myself, are really tremendously excited about. So Steve.
Awesome. Thank you very much for inviting me to talk today. And we'll kind of go through the acute kidney injury adverse event definitions. As Ryan mentioned, I had an incredible group to work with. So I'm speaking on behalf of Aisha, Warwick, Catherine, Marlise, and Matt, as we kind of go through the AKI adverse event definitions. We have three major ones that I really want to review, two of which we're going to review a lot more in depth. So the existing definitions of AKI in patients on ECMO or mechanical circulatory support. Right now we have the MCS-ARC definitions, we have the ELSO existing definitions, as well as an operationalized definition that the KIDMO group used. So the MCS-ARC definition really focused on the adult population on cardiac support, and our ELSO definition really encompasses all ages for all indications in patients on ECMO. The KIDMO definition really aims towards the neonatal and pediatric patients less than 18 years of age for all indications of ECMO. This is the one we're not going to go very much in depth because it uses the KDIGO creatinine-based definitions, which we'll get into, but it doesn't include all of the definitions that we want to propose. And so we'll see in the future how we advance this operationalized definition. First, evaluating the current MCS-ARC definition. As we see, the MCS-ARC definition really chose to use the KDIGO framework as a backbone for their definition. The KDIGO AKI framework was put forth about 10 years ago. It really harmonized the existing definitions of AKI, such as rifle and the Aiken criteria, and combines a creatinine-based definition as well as a urine output definition, which are really functionally very easy at the bedside measures and proxies for GFR. Further, the MCS-ARC definition really includes staging criteria, which we can see, as well as a framework for chronicity. So at the bottom, we'll see kind of that chronic renal dysfunction. Some of the really good things about this definition is that it does use the established staging criteria, and it does use and alludes to chronicity. And it can also be used for adult and pediatric populations. Unfortunately, it doesn't really include a timing element, so to help kind of attribute that kidney injury to anything. And it doesn't really apply well in the neonatal population. Importantly, all CRRT applications are also included. For instance, if hyperammonemia were treated with CRRT while on ECMO, it would be included here. Next, reviewing the ELSO definition. Here, we can also see that it has a staging criteria and a really important piece that the MCS-ARC doesn't have, which is this newly acquired creatinine. Again, all RRT for all reasons is included here. And so the good things about the ELSO definition is that it had that staging that we talked about. It also includes a timing element, the newly acquired creatinine while on ECMO. Unfortunately, it doesn't use the modern creatinine-based definitions the way that the MCS-ARC definition incorporates the KDIGO stuff. It also doesn't include a urine output definition. And again, all CRT applications are included. So from here, we really expanded out and looking at the literature as a whole, and we focused on the existing definitions and availability to implement them. We started with the KDIGO definitions, which kind of includes adult pediatric patients and uses that serum creatinine and urine output based definition, as well as just stage severity. It really is considered the gold standard of AKI definitions in the literature. We also looked at Podium definitions, which included critically ill children with multiple organ dysfunction syndrome. And really that renal dysfunction is one of those organs. It uses what I'll call a conditional KDIGO criteria and incorporates and introduces new ideas like what does fluid balance do here, and really establishes a framework to establish kind of baseline creatinine, which is important. The ADKey 16 group, as well as an advanced KDIGO group, thought about how to use creatinine-based timing and incorporate the duration and endurance of an AKI event, really from this continuum from an AKI event to acute kidney disease and chronic kidney disease. And then we'll go over the AWAKEN criteria, which included critically ill neonates and operationalized a modified neonatal KDIGO criteria. Looking at the KDIGO criteria, here I put forth again as kind of the gold standard in the literature. It's really important here that we have inclusion criteria really for what defines AKI. This is here at the top. Importantly, we have creatinine and urine output, as well as this timing component within 48 hours or in the prior 7 days, which really gets at the acuity of the injury that's occurring. From here, once one has AKI, we can stage it. And so we get broken down into severity staging. This is really important because validation cohorts have really demonstrated the importance of both serum creatinine and urine output staging with their associations with outcomes, including length of stay, mortality, and a variety of populations. Here, the Podium criteria put forth a framework for renal dysfunction in that context of multiple organ dysfunction. It uses the KDIGO urine output and creatinine framework, but importantly, it doesn't really apply a severity grading to this. But some new elements that are really important that we ought to think about are the conditions for CRRT here are important, and really the initiation for any reason other than toxic ingestion or hyperammonemia, and really getting at CRT for kidney injury related conditions. Further, it introduces this idea of a fluid balance greater than 20%, as there's some evidence that this threshold, and probably even much lower, is really associated with poor outcomes. One of the most important elements of the Podium definitions is that it put forth a baseline creatinine definition. Here they operationalize the idea that a baseline creatinine is the lowest measured in the 3 months before admission. However, unlike adults, many children do not have previously measured creatinines, and therefore, when unavailable, they put forth this age and sex indexed and validated criteria for reference as baseline creatinine criteria moving forward. These values were derived from healthy children, but have been validated in critically ill pediatric patients. The next two groups that I want to talk about really are getting at the same idea, which is this continuum from AKI and acute kidney disease, as well as chronic kidney disease. ADKey 16 put forth and highlights the really important outcomes that are associated with patients with early, late, and non-recovery of kidney function after an AKI event. The ADKey 16 group proposed a timeline of these definitions, suggesting that AKI exists in a time frame less than 7 days, and afterward it becomes an entity called AKD until 90 days and CKD thereafter. A group led by Sue operationalized this definition in 2020 in 168 adults who received ECMO for more than 7 days, and outcomes were able to be followed for up to 10 years, and really did demonstrate that the AKD after AKI was associated with increased mortality compared to no AKD after AKI after support with ECMO. KDIGO put forth another kind of framework for AKD and CKD in 2021 that really is reminiscent of that ADKey 16 group. And this framework really indicates that AKD is a large umbrella that includes not only AKI, but other acute kidney diseases such as glomerulonephritis that might not meet AKI criteria. But here we can see reference to the resolution and the importance of the endurance of an AKI event or a kidney dysfunction event. So here again, the AKD umbrella is anything that happens within 3 months, and anything that really meets AKI criteria or other dysfunction criteria after 3 months really becomes this entity of CKD. And last but not least, the neonatal population really does need to be addressed. Here we can see a familiar table from the Awaken group that really lists our serum creatinine and urine output measurements along with staging criteria that is really familiar at this point. However, there's really important deviations for neonates that really speak to and relate to the nephrogenesis that occurs throughout early development. First, urinary output here is recorded over 24 hours as opposed to those 6 to 12-hour increments. Second, in order to classify a patient with serum creatinine defined AKI, each measurement was compared to the lowest previous measurement to detect both an absolute and a percentage rise from whatever that previous baseline was. Comparison to the lowest previous serum creatinine was operationalized in this, and that value is really necessary because serum creatinine values normally decline over the first few weeks after birth, such that the baseline value is constantly changing. And so the lowest previous one serves as an internal baseline for each individual patient, and we can use kind of indexed to that patient relative rises. Third, a serum creatinine cutoff of 2.5 was used rather than the 4 that was used in adults. Here it's used 2.5 for stage 3 for neonates, since the cutoff of 2.5 references a glomerular filtration rate of less than 10, which would be the equivalent of 4 in adults. And bringing this all together, this brings us to our draft definition of acute kidney injury adverse event while on ECMO. And this draft definition really includes a lot of the elements you've seen already. First, we really wanted to create this inclusion criteria and then create that staging criteria that really harmonizes with other definitions. So here in this dark box, we can see that AKI is defined as any of the following that occurs during ECMO support. So we have an increase in creatinine more than 0.3 over 48 hours, or more than 1.5 times baseline over 7 days. We also have urine output criteria that is both referencing for pediatric patients as well as neonatal patients. And we have this idea of initiation of renal replacement therapy only for the treatment or prevention of complications of acute kidney injury. These definitions really reference an acute increase, and baseline will be defined as similar to what Podium put forth. The lowest creatinine measured in the previous 90 days before admission, and if absent, the baseline will be age and sex index based on those tables from Podium. In neonates, we'll operationalize the baseline being the lowest creatinine in the previous 7 days, same as our colleagues who were operationalizing the neonatal KDIGO criteria. And for renal replacement therapy, that treatment or prevention of complications of kidney injury really does exclude CRRT for those kind of toxic ingestions or hyperammonemia. And it also mitigates classifying CRRT use for a center's standard ECMO setup and ECMO circuit. While we know that survey data from ELSO centers really suggests that fewer than 5% of centers use CRRT as a standard setup with their ECMO circuit, it is possible that protocolization may increase uptake of this practice, as we wanted to make sure that CRRT indications for kidney injury or complications of kidney injury were able to be referenced. From here, we can stage both based on severity as well as timing. And so here, our stage 1, stage 2, and stage 3 criteria are similar to the KDIGO staging, as well as the modified neonatal KDIGO. And we also have this reference to the timing severity, so the duration and endurance of an AKI event matters. And so we have acute kidney disease, which is an AKI event that lasts up to 90 days, but resolves before 90 days. Or chronic kidney disease, which really is an AKI event that persists for more than 90 days. One thing that's notably absent from this definition is the lack of other biomarkers, non-serum creatinine or urine output type of biomarkers. And while there may be better injury and filtration markers like NGAL or cystatin C, they have yet to be validated in this population as definitional. And so until then, we're using what we've been able to validate, which has really been this KDIGO framework and applying it to our pediatric and neonatal populations.
Thank you so much, Steve. I wonder if you can just pop the yes, you've done this before, and then we'd love to hear questions or comments from anyone who have questions or comments.
I have a question. Thank you, Stephen, and the people you did this with. I think it's amazing. You did go into it a little bit, but there are centers, like you mentioned, that use this CRRT electively. Do you think we need to exclude them then in the future because this is for research purposes? Because using the CRRT electively will influence the other parameters that you've defined in here, like urine output and creatinine rise and all that. And by eliminating them, that might be doable. The only question is, if you look at the ELSO registry, we do not register whether or not somebody puts CRRT electively or not. So how do you propose we deal with that subtype of those groups that use it electively?
Yeah, Peter, that's a really great question. It's something that we've wrestled with a lot. And one of the things that we know is, again, I referenced that fewer than 5% of ELSO centers will do this, but the opportunity exists in the future. One of the things that we know is that somebody who is put on CRRT for non-kidney injury related reasons can still develop a kidney injury. And so they can still meet urine output criteria after that. And they can still meet creatinine-based criteria. And so if the patient is put on for non-kidney injury related reasons and develops a kidney injury, it would still be considered a kidney injury adverse event. But it wouldn't be necessarily included as the RRT indication. It would be for serum creatinine or urine output based indications.
Thank you.
Peter, it looks like there's a question in from one of the attendees. Are you able to see that?
I don't see one in the there are no open questions in the Q&A. Maybe
actually,
in the
I
chat.
can see now. I think Jonathan Smith has his hand up.
Ah. John, are you able to pose a question, or otherwise put it in
John won't be able to place a question. I have to change the setup, but he can write down his question in the Q&A.
Please, John, and then I will. Some of us will relay it. Yeah.
Thanks. That's actually a really good distinction. We've scheduled in a lot of panel discussion here, and so I think one of the things, as we're going through the talks, if you can, please just type it into a question, and so that we're able to kind of have those coming along, and then that way we can read them off. But we really very much want the feedback and want the interactiveness of this. So thank you.
We were really happy. I'm sorry, go ahead.
No, I think it's come in. So John asking, the units for creatinine are wrong in the definition, he believes. So that maybe that needs some attention. Units for creatinine. Yeah, so we've got different definitions, of course, for the units across the globe. So this is a global initiative, so we'll need to make sure that all the units are correct for the different countries out there. I think that's what he means.
Perfect. I appreciate that. I will make sure that gets addressed.
I will say, Steve, you're using the numbers that are used in the, I think, which is also the I and KDIGO, I think, right, is also about that international scope as well. Is that right? And so we definitely want to make sure that this platform is able to be used across. So we'll definitely turn attention to that. We're also trying to maintain some harmonization with some of the existing elements that are out there. So thank you. Really appreciate that, and want to continue to hear things like that as well.
Yeah, it says millimoles per liter, but it should be micromoles per liter. I think that's the basic difference.
Thank you.
I don't see any other questions coming in at the moment. The other panelists have questions for Steve, and otherwise we'll proceed with the next definition. Is that okay with you, Ryan or Peter?
Yes, of course. Peter, I think the other thing you're getting ready to say is if it isn't difficult to change the setting so that the folks are able to speak up in the middle, is that where you're going? We're happy.
However that works, we would certainly be happy to hear from other people. But the other way we can hear from people is that at the end of our presentation, we will have a QR code with the Survey Monkey, and we can hear anything. We're happy to hear anything, any feedback from you by the Survey Monkey as well. So next up on our list, we have Ariane Willems, who's going to talk to us about the bleeding definition that was developed. Ariane is a fantastic collaborator, and I've been very lucky to work with her on other projects and was delighted that she accepted our challenge to join us for this one. Ariane joins us from Leiden in the Netherlands with Pete, and where she works as a PICU attending, and she has expertly led quite an experienced group of investigators to help us bring together a bleeding definition. So Ariane, when you're ready, if you can share your slides, we have really appreciated your leadership in this space. Thank you.
Ariane, I think you might be muted.
Sorry, I'm muted.
Yes.
Sorry. So thank you very much for your kind introduction, and thank you to invite me to lead this amazing group of people. We try to have a good bleeding definition. As Alain Combs told us, it's necessary, but it's also challenging. Okay. A very brief introduction. As you know, bleeding remains a frequent and significant complication in children on ECMO. Some publications, and it depends on the population studied, but show that up to 70% of the patients are bleeding, and that the median amount of blood lost during a run is 120 milliliters per kilogram. Both events, so the fact of bleeding, but also the amount are independently associated with increased risk of death. The causes, you have patients' blood that is interacting with the foreign surface of the ECMO circuit that activates the inflammatory and hemostatic system, causing a coagulopathy. Therefore, you need systemic anticoagulation, but systemic anticoagulation is also associated with bleeding, and in some cases you have an intervention to cannulate a patient, but a lot of patients receive ECMO after a surgical intervention, who is also a risk factor for bleeding. Interestingly, the severity of bleeding can be characterized on two ways. You have the amount of blood lost and its physiological consequences, but also small amount of blood in some particular parts of the body can be very severe and life-threatening. This is a recent systematic review on coagulation in pediatric ECMO patients. And what you see is that you have as much definitions of bleeding as you have studies. So there's an urgent need for a standardized definition of bleeding. However, I can assure you it remains very challenging. And why is it challenging? Because when you are bleeding, you have different consequences, and you can choose which item is the best to define bleeding based on these consequences. The first consequences we all know is a loss of intravascular blood volume. So you lose volume, and that leads to hemodynamic compromise. And as you say, defining hemodynamic compromise in ECMO patients, it's not very easy. The second consequence of loss of blood is the loss of red cells, and therefore the oxygen delivery to tissues. And finally, you also have loss of platelets and coagulation factors who may cause or worse or further worsen a coagulopathy. You can also see a bleeding on the way you respond to bleeding. First, as a response to bleeding and to the intravascular volume loss, we give fluids and blood products, and you can measure how many milliliters per kilogram fluid and blood products you give to the patients. You can also look at what actions were needed to control the bleeding. You can act on the coagulopathy by giving blood products, antifibrinolytics, hemostatic agents. You can also, especially in ECMO patients, decreasing, stopping, or even reverse anticoagulation, because as you know, it is one of the causes of bleeding. And finally, in some situation, a surgical intervention is needed to control the bleeding. Finally, bleeding has also consequences on patients' outcomes, such as multiple organ dysfunction syndrome and death. But it's difficult to define in our ECMO patients because they are very ill and all have some way of organ dysfunction, because those are the sickest patients of our ICUs. As you imagine, all these items have advantages and disadvantages, and we think as a group that the ideal bleeding score should include first physiological variables that are appropriate for the studied population, in this case children on ECMO, should include sites of possible bleeding that are particular to the population or intervention studied. We think that the score should be untied to interventions, because all those interventions are physician dependent, subjective, and highly variable. Each center has his own thresholds for transfusion of blood protein, has its own protocols, and it makes it difficult to introduce this variability in a definition for bleeding. And finally, our score should be validated against meaningful clinical outcomes. So there are a lot of definitions, and we retained 4 definitions to build our bleeding score: 3 in mechanical circulatory support patients, and 1 in critically ill children. I will go through the 4 definitions. So the first definition was presented also for the acute kidney injury group, and is the MCS-ARC definition. Here you can see the definition base for the bleeding adverse events. It was published in 2020 by Kormos and al. It's a definition who is based on severity. You have a type 1 who is defined as minimal bleeding, and a type 5 who is defining fatal bleeding. One of the exceptions is the type 4, who is a bleeding that is immediately related to the implantation of the VAD. As you can see, this definition considers different aspects or parameters of bleeding, and one concept that you don't find in other definition is the concept of action or not action associated with the bleeding. In the type 1, you have not you need no actions to control the bleeding. They also use the hemoglobin drop, who is a surrogate marker for blood loss, for the loss of the red cell mass. And finally, they also define critical sites of bleeding, such as you see in type 3, you have cardiac tamponade in the type 3B. As I told you, type 4 is a peri-implantation bleeding, and there they also consider the amount of chest tube output and the amount of red cells transfused to define bleeding. Interestingly, they in this publication, they also think it's important to describe the location of the bleeding based on the frequency associated with here the intervention or the population, being mediastinal, thoracic, lower GI, chest wall and upper GI bleeding, and the moment of occurrence, because it's most of the time associated with the cause of the bleeding. The second definition we relied on to build our definition is the EuroELSO definition. And here you can see the definition. It is a yes-no definition, so not based on severity. The definition is based on the need or not of a specific amount of red blood cell transfusion or whole blood transfusion, or the need for an intervention. And you have also a detailed definition of site-related bleeding, such as peripheral cannulation site bleeding, mediastinal cannulation site bleeding, and surgical site bleeding, and also GI hemorrhage. The third score we use to build our definition of bleeding is the basic score. It's a score, as I told you, in critically ill patient, and it was published in 2019 by Marianne Nellis and colleagues. It's a bleeding score in critically ill patient, but it is also studied in cardiac surgery and ECMO. It's a consensus-based definition of clinically relevant bleeding. And there, the items used to define the severity score, and you have three types of bleedings. You have minimal bleeding, you have moderate bleeding, and you have severe bleeding, but also, as you see, a definition for fatal bleeding. And the items used to define bleeding are first worsening multiple organ dysfunction syndrome, based on the PILO2 score criteria of organ dysfunction. You have also a hemodynamic compromise and defined by heart rate or blood pressure. You have loss of red cell mass, and so a definition of severity based on hemoglobin drop, but also a definition based on how the amount of blood lost and site-related bleeding. As you can see, you have in the severe bleeding group, loss of intraspinal bleeding, intra-articular bleeding, and intraocular bleeding as severe sites of bleeding. Interestingly, in this definition, you can see that they don't use interventions and actions needed to control the bleeding to define severity. And then finally, you have a recent published consensus statement published in 2022 by Levy and colleagues. It's a definition in different settings, so you have different definitions in cardiac surgery and in the perioperative period compared to ECMO and VAD patients outside the perioperative period. It's a 5-point ordinal scale based on severity. And there, the items used to define bleeding are transfusion of blood products, but not only red blood cells, but also other blood products such as plasma and platelets, the administration of antifibrinolytics and hemostatic agents. And interestingly, you have changes in anticoagulation targets or stopping or reversal of anticoagulation as an action to control bleeding. They also define critical bleeding sites. Here you see the definition related to cardiac surgery and perioperative ECMO. You see that you have 5 types of bleeding with who are defined based on anticoagulation, changes of target or doses, and the localization of the bleeding, but also the amount of transfusion. Those are the definitions of ECMO and VAD outside the perioperative period. So we drafted our definition, and we choose a severity-based definition, going from type 0 to 4. And the most easy to define were type 0, no bleeding, and type 4, fatal bleeding. And we retained 2 parameters. The first one was the amount of blood lost, or the hemoglobin drop, which is a surrogate for the amount of blood loss, knowing that you have some confounding in ECMO patients. We also defined critical sites, but we thought it was important to define bleeding related to the physiological consequences on hemodynamics and oxygen delivery defined by lactate level. So the first 2 types were type 0, or no bleeding, no signs of hemorrhage. The second type was type 1, also called minimal bleeding, and is defined as any one of the following. So it's a measurable blood loss of less than 2 ml per kilo per hour, or bloody dressings change less than every 6 hours. Clinically insignificant bleeding from the endotracheal tube, nasogastric tube, mucosa, or the head, such as petechia and purpura, or action or bleeding not leading to actions except observation, the use of local hemostatic agents. The type 2 or moderate bleeding is defined as overt bleeding associated with any of the following. First, an amount of blood loss of 2 to 5 milliliter per kilogram per hour for 2 hours or more. A hemoglobin drop of 3 grams per deciliter over less than 12 hours, but with no impairment of oxygen delivery capacity, and with defined no impairment in the oxygen delivery capacity by lactate below 3, or in patients who start with the lactate above 3 before the bleeding, that the lactate is falling over the last 12 hours. We also defined moderate bleeding as GI bleeding, pulmonary hemorrhage, mediastinal cannulation site bleeding, or surgical site bleeding that has a physiological impact but does not meet the criteria of severe bleeding. Then the type 3 is major bleeding, and we defined it as overt bleeding associated with any 2 of the following. This means an amount of blood loss above 5 milliliters per kilogram per hour for 2 hours or more. Bleeding that is associated with hemodynamic compromise in absence of other causes. And we suggest to define it with the need to increase vasoactive agents. And we think it's difficult to define it in VA ECMO. So we retained it only for VV ECMO patients. Or we also defined hemodynamic compromise by the need to administer volume to maintain pump preload. Major bleeding could also be defined based on severe hemoglobin drop above 3 grams per deciliter within 12 hours, or impaired oxygen delivery capacity, evidenced by a lactate above 3 and increasing in 2 consecutive measurements, or solely a lactate level above 5 millimole per liters. This definition comes from the titer trial in children for ECMO. And finally, we think that bleeding that causes a compartment syndrome in the abdomen or in a muscle compartment is also a reason to define it as major bleeding. Finally, the type 4 is a fatal bleeding, and we defined it as a bleeding that is the cause of death, as in the MCS-ARC definition and defined by Nellis and al. I thank you for your attention, and I'm happy to hear your comments and suggestions.
Thank you, Ariane. Again, we're really interested to hear people's perspectives on the bleeding definition, particularly. I don't think you've got a summary slide that would be able to leave up as people are considering the questions. Ariane, no. I think that's
maybe I can put on the major bleeding definition.
That might be great if you don't mind.
There's one question. Somebody asks for the definition of bleeding, if it can be applied to adult ECMO too, and in which way does this definition relate to the adult definition, or is that something completely different?
Oh, thanks. It's an interesting question. The big difference with adult definition is that in adults you don't define bleeding in milliliters per kilogram. It's always based on a volume, because you don't have this wide range of weight in adults. So that's one of the difference. But I think a lot of items can also be used in adults too. We used the hemoglobin drop of 3 grams per deciliters. We extracted this from the MCS ARC definition. That's one of the examples.
So we can use these in our teenagers as well.
So yes.
Yeah.
Yes. We try to be to have a definition for a huge group of neonates, children, but also adolescents and teenagers.
Yep.
One of the things that that became very clear to us as Ryan and I did engage with stakeholders, Dr. Kuhn put us in touch with many of the trialists in Europe, as we mentioned earlier, and the current definition that's being used in adult trials for bleeding is actually most commonly the bleeding ARC definition. It's called BARC. And it was on one of our slides earlier. And so this definition was built out of the BARC definition specific to pediatrics. But I think that in terms of would this be applied to adults, they'd probably be more likely to stick with the BARC definition. But hopefully an adult process will follow this pediatric process, and we can remain harmonized across the groups.
Okay, thank you. Another question came in from Mark Davidson. So I don't know how, no, let's not go there. The question he poses is, I think, a correct one. He says there's a definition here about the vasoactive instability, that the need to increase vasoactive agents in VV ECMO. Not all children are on vasoactive agents on VV ECMO. So the word increase might not encompass at all, and we may need to use start vasoactive agents, or maybe other considerations, more volume required. So any comments on that? Is that trigger increasing vasoactive enough? Is it appropriate for pediatric cases is the question. So
yes, you can also, I think we can better define it, but you can, when you start from 0, and you can also
increase
vasoactive agents, but I think we can better wording it.
Yeah. I don't know if that answers his question, but otherwise we'll hear back.
But it's difficult in ECMO to define hemodynamic instability or compromise and what you are doing, because when a patient is bleeding, you are doing a lot of things, and patients on ECMO can be stabilized, but they can only also be unstable for other reasons than for bleeding. So it's difficult and challenging to define hemodynamic compromise in this population. But thank you for the comment. We will.
He's happy with the answer.
Rephrase.
He understands it's difficult. I have another question from the audience about intracranial catastrophic bleeding. It might not cause death per se, but it would probably lead us to rethink the ECMO run, and it's not, you know what I mean? So that we will redirect care, and the patient might pass away anyway. Would this be type 3 or type 4? These are things that we need to define properly beforehand.
Yes. So maybe you, Ryan and Peta, can add some comments on that after what I say now. But we decided not to define intracranial bleeding because it's defined in the neurological adverse events of ECMO, and we didn't want that you had two complications for the same problem. You know, I think you can better explain it than me, Peta or Ryan. But that's why we didn't. But we are aware that one of the causes of one of the severe complications of ECMO are intracranial bleeding. But one point is the pathophysiology. You never know if it starts with a thrombotic event and you are bleeding secondary, or if it's a bleeding on its own. And secondary in this process, it is the neurologic adverse events group who is defining intracranial bleeding.
Yeah, I think, Ariane, I don't have a lot to add to the first part of what you said. I think that's spot on is what we're thinking. Happy to hear challenges to that. I think these are important opportunities to really think about this and wrestle with this with the community. But I think the other thing that I heard a little bit in the question was, 3 versus 4, because maybe the bleeding event itself didn't lead to the patient's immediate death, but rather the consequence of the bleeding event changed the goals of care, and the patient was redirected as a consequence. And that's not actually something I've wrestled with in my own head. And so I really appreciate the opportunity to think about that. So that's a really, really helpful question. I think my first response to that is probably to say that if the bleeding event led to a redirection of goals of care and the patient were to die, I would probably lean towards calling that a fatal bleeding complication. But I understand why that may not be an appropriate aspect. And so we definitely want to kind of socialize that with our team and kick that around. And if the commenter has, an opportunity to, offer any ideas, that we'd love to hear those as well. I think the other side of that coin as well is just that we in the neurologic events scale out the bleeding consequence, or sorry, not the bleeding, the neurologic adverse event consequence. And so, what impact that has on the neurologic functioning would be captured there, which is maybe another reason why that bleeding event being captured in that for neuro makes sense. So thank you. Really, really helpful question. Very much so. Any other questions? I just saw our Q&A cleared out, which is, I think, partly because we at the time to move on to the next talk, but also want to make sure that there's nothing else that's burning in the audience before we do shift gears. And Peta, if you could introduce the next when that time comes.
So if there really are no other questions, I will just take a minute to thank the other topic leads who aren't actually presenting today. We spent 12 hours over a 2-week period discussing these adverse events. And with 2 hours today, we took the opportunity to highlight 4 topics that we could explore and get your feedback on. But we had a number of other leaders of expert groups, including Dr. Erica O'Neill, who's in the US military and in San Antonio, Dr. Ahmed Saeed at the University of Washington, who led a number of different adverse event definitions actually, and Dr. Giulia Benedetti, who's a neurologist, neuro-ICU person who led our neurologic definitions. And we really appreciate all of the investment of both the leaders as well as the experts that contributed to those not highlighted today. Dr. Mark Anders and a group also put together a scoping narrative literature review to ensure that we'd captured all of the adverse event definitions which may inform the groups. And so there's been an enormous amount of work that's gone into the background of what you're seeing presented here today. So special thanks to all of those people as we move on with our hemolysis definition. So Ariane, you can stop sharing your slides if you don't mind. and I'll ask Dr. Francesca Sperato to start sharing her slides while I give her a quick introduction as well. Many of you will know Francesca, who, as you know, is a pediatric intensivist who trained in Padua in Italy before moving to Boston, where we're delighted and hoping that she will stay. She's training with us as a cardiologist. In addition to her background as a pediatric intensivist, she's a colleague and a friend, and I was so pleased when she accepted our challenge to help us lead this group to define hemolysis as an adverse event. She was also hugely contributing to the new definition of right heart failure, as well as left atrial hypertension. So we're very much indebted to Francesca for all of her work. Take it away when you're ready, my friend.
Thank you so much, Peta, and thank you for the opportunity to present this topic today. I'm presenting the hemolysis adverse event definition on behalf of this incredible team that you see here listed, and I would like to thank each of them for the amazing contribution that they provided for this definition. As you know, hemolysis on ECMO is a real challenge, and it might be patient-related, circuit-related, drug and management-related, and oftentimes require action. So it is really important to have clear criteria and to recognize hemolysis in time. Existing definition of hemolysis in patient on ECMO so far are mainly two: the MCS ARC definition, and as you know, they refer mostly on adult population, on cardiac support, on durable VADs, with some reference to also temporary VAD, and the EuroELSO registry definition, which refer to all ages, to patients supported by ECMO for all indications. In this diagram, you can see very nicely summarized the definition of hemolysis based on the MCS ARC adult consensus statement. And as you can see, this hemolysis definition is met if you have at least one between a plasma-free hemoglobin more than 20 milligram per deciliter, or an LDH more than 2.5 times the upper limit, with a flag for this LDH because the group was recognizing that there are multiple confounders also for the adult population, especially laboratory error, hepatic or pulmonary dysfunction. And so LDH should be taken into consideration for hemolysis only if confirmed and all the other confounding are excluded. Hemolysis is also defined within this consensus in minor hemolysis, where when there are no clear symptoms, or major hemolysis, when we have either hemoglobinuria, anemia, hyperbilirubinemia, especially with predominantly indirect bilirubin, or a clear pump malfunction. This is the EuroELSO registry current definition, and as you can see, we have two grades as well, but they are instead of a minor and major, we define it moderate and severe hemolysis based on two cutoffs. So moderate hemolysis is defined as a peak plasma-free hemoglobin between 50 to 100 milligram per deciliter, occurring at least once during the ECMO run and sustained for at least 2 consecutive days. And severe hemolysis is defined as a plasma-free hemoglobin more than 100 milligram per deciliter, or occurring at least once during the ECMO run and sustained for at least 2 consecutive days, or if the hemolysis entails a change of one of the major components of the circuit. For completeness, we have also reviewed the literature for other existing definitions, or and we didn't find any in particular for pediatric patient on ECMO, or for other pediatric non-EuroELSO based a definition of hemolysis. So in these tables, you can see summarize the studies that have clearly defined hemolysis. There are other studies that use the plasma-free hemoglobin the peak plasma-free hemoglobin as a continuous variable, so they are not included. But among studies that clearly define hemolysis as an event, so a binary outcomes, you can see here that they are mainly retrospective and single centers, and they are all using the plasma-free hemoglobin as a biomarker. Very much consistently, they identify severe hemolysis as a plasma-free hemoglobin more than 100 milligram per deciliter, while cutoff for moderate hemolysis are a little bit more variable, and they go between 40 to 50. And some of them, they don't really grade the hemolysis, and they just have one cutoff. I also added, we also added two more columns here to point to your attention on the fact that neither of these studies use other biomarker, especially LDH, for the definition of hemolysis. That is thought to have a lot of confounders, especially in pediatric age. And also there is a not clear time for the hemolysis assessment. So some of them entails assessing hemolysis when clinically indicated, some of them daily, and some of them only occasionally once a week. And I'm referring, I'm presenting these two particular details of these studies just for a comparison with the adult ARC MCS definition. This table is a very nice table from a review by Defer et al. In 2020, and summarized very nicely the dimension of the problem. As you can see, and I'm going to introduce in my slide the unit of measures that we have used so far, just for facilitate. And as you can see, the severe hemolysis among pediatric populations supporting either VA and VV ECMO range with a prevalence between 7 to 43%, while moderate hemolysis between 12 to 23%, and mild hemolysis can be present up in 84% of patients. And just to put everything in perspective, the two cutoff that has been chosen so far from the two definitions that we have just reviewed are the ALSO definition, the 50 milligram deciliter cutoff, and the MCS ARC definition chose the 20, so a little bit lower cutoff. And this second table, very nice as well, summarize the association between plasma-free hemoglobin or severe hemolysis, mostly severe hemolysis, also moderate hemolysis, with the outcome. And they found both in pediatric and adults, they found clear association between a peak plasma-free hemoglobin, moderate or severe hemolysis, and either acute kidney failure or mortality. And just for completeness, we also reviewed the literature in terms of other biomarkers, and this very interesting study evaluated mainly LDH, AST, and ALT, and gross hemolysis as other markers of hemolysis, comparing it to the plasma-free hemoglobin. And they found that mainly the LDH and the AST in absence of ALT can be considered as a surrogate of hemolysis. But I would like to point out as the correlation between, in particular, plasma-free hemoglobin and LDH is a moderate correlation, and the authors themselves commented how there are so many confounders for the LDH in pediatric age in particular. So we are presenting now our draft definition based on all these evidence we have reviewed. So we would like to use degree for this definition, and so we We recalling the ECMO existing definition, we have defined a type 1 or moderate hemolysis and a type 2 or severe hemolysis. We briefly took into consideration also to add a mild hemolysis, but in the effort to harmonize with the pediatric ARC-MCS definition, we decided to not include it. And so we our definition so far include a type 1 moderate hemolysis defined as a peak plasma-free hemoglobin between 50 to 100 milligram per deciliter occurring at least once during the ECMO run, sustained for at least 24 hours, or if we have only one measurement, if the hemolysis entail a major circuit component change. And type 2, a severe hemolysis, as when the plasma peak plasma-free hemoglobin is more than 100 milligram per deciliter, occurring at least once during the ECMO run, sustained for 24 hours, or similarly to the moderate part, if we have only one measurement, more than 100, but we need to have a major circuit component change. We decided to not include LDH because of the evidence of a lot of confounders in pediatric age and on ECMO. But we did feel the need to add a flag for patients post-cardiac surgery because they underwent those that underwent a cardiopulmonary bypass surgical intervention, because the hemolysis in that case could be related also to the cardiopulmonary bypass run itself. So a flag will be added if hemolysis criteria are met only within 72 hours of cardiopulmonary bypass run. And I'll let Peter, Ryan weigh in for any additional thoughts, and all of you for any questions and suggestions. Thank you.
Thank you, Francesca. That was fantastic. I really appreciate all the work that went into this. I don't have anything to add to your wonderful presentation, but yes, if there are questions, would love the opportunity to address those.
I don't see any questions arising. Aparna, do you have a question?
No, I just wanted to say a very clear presentation by Francesca, and I think one question that I had in mind was that in general, when patients are on ECMO, how often is the plasma-free hemoglobin measured? Now, there are some institutions who do it on a regular basis. Some would do it if it is clinical, and from what when I look at this, I just get this feeling is that if you're saying that even if it is a one-off measurement, and it is high, and it is sustained for at least 24 hours, then it would be either type 1 or type 2. So it's just that, here, if you search for it, you may find it more than you think that it's there, and whether it's clinically relevant or not will be guided by whether you needed to intervene. So how would you say in terms of when you're planning any trials based on this definition, how often would you monitor plasma-free hemoglobin?
And thank you, Aparna. This is a great question, and we talk a lot about this timing of measurement. And I think what the rationale why we didn't explicitly ask for a timing of assessment is because we felt that like major hemolysis as a for example, you can see in the adult MCS ARC definition, have usually signs and symptoms, and it's a clear endothelium dysfunction. So we expect that the patient would be sick, and we expect that the team would start investigating the patient. And so among the panel of the labs, like looking for hemolysis based on, you know, clinical signs and symptoms would be something that is very often done. But I agree, like this definition needs a clear test to be sent. And also some of the centers may not have peak plasma-free hemoglobin. So, but on the other side, and so this is part of the challenge of this definition, but on the other side, we really needed to have something very clear and precise to compare this event to other centers. So yeah, we elected to leave it as at least one measurement that needed a ECMO circuit change or 2 measurements within 24 hours. I don't know if Peta and Ryan had anything to add to that.
No, Francesca, I completely agree. I think we are talking about this in the context of clinical trial adverse events specifically. And so depending on the investigators, they can choose to request plasma-free hemoglobins on a daily basis or otherwise, but that would be part of the clinical trial design. Our definition is only about the duration that we'd expect to see it there before the definition was met. I'm just looking at the chat. Do you mind if I step through 1 or 2 of the other questions that have been raised, because it's fantastic to see this level of engagement with the definitions. Many of the questions in the chat were actually things that were discussed at length in the adverse event definition meetings that we had, particularly in regard to the availability of plasma-free hemoglobin at all centres and the potential for other biomarkers to be subbed in or added to the definition. There was a lot within the community. There was a really strong preference not to use less specific biomarkers, for example, LDH in the ECMO population, the way it's being used in the MCS population, particularly because ECMO patients tend to have multi-organ dysfunction, and the use of the less specific biomarker was really problematic for our population. A number of our experts were quite invested in the carboxyhemoglobin question that I can see Alexis has asked about in the chat. Our reason for not including carboxyhemoglobin in the definition at this stage is because there aren't equivalent studies across the board that would help us differentiate according to levels. And it really doesn't appear to be established in the same way in the ECMO population, although it is probably coming. And so our adverse events, according to the ARC process, are expected to be reviewed every 2 years. And so if there is evolution in the available data to potentially include carboxyhemoglobin in future iterations, then we're really hoping that gets there. But just being able to identify at this stage that that we weren't able to find other definitions that include carboxyhemoglobin for hemolysis meant that we did not keep that in the definition as it stands. And I think the other question in the Q&A, Francesca, I'm sorry, I'm stealing all your thunder here, is just about the potential confounders to plasma-free hemoglobin measurement. And one of the things that I think we should do in the context of the specific question from Robbie about lipid emulsion-based therapies is add a part of the definition that highlights the importance of excluding other confounders to the test result. I think that should actually address that really well, but thank you for raising the issue.
There's also another question about what is the consideration if patient is on tandem CRRT and ECMO regarding hemolysis. Francesca, do you want to take that question?
Yeah, those are all great questions, and we went through all in our discussion. I think if you have a CRRT, your hemolysis is going to be even higher. So of course, that in that case, it can be related for both the ECMO and the CRRT. So we thought that it was okay to refer these adverse events to the ECMO itself. Peta, anything to add on that?
No, only that the definition of hemolysis stands, the epidemiology may be slightly different, but we would be comfortable with the patient level definition being the same.
Peta, I think this also might be an opportunity to introduce an adjacent concept, and then maybe I can switch over to introducing Arbor to talk about devices. But I think that one of the things that Peta pushed us to think about carefully was this idea of a supplemental table, which is really around thinking about if you were designing a clinical trial for these adverse events, are there other aspects of the epidemiology that might inform maybe the way that you stratify the randomization process. So there are a number of examples where this might be important. I think maybe key to hemolysis is that, if you had cardiopulmonary bypass coming in, that also would perhaps upfront impact what your hemolysis was coming off, and wanting to think about that. And so that's another element that we're not presenting here. But also you could make sure, the use of CRT on ECMO may not be something that you could anticipate ahead of time, but it's something that you could certainly make sure in your randomization that you had balance amongst in terms of looking at that table 1. And so I think is really important. And so up next, we have Alvaro, who is been a fantastic collaborator, and just really appreciate his willingness to lead us into a lot of uncharted territory, really, with defining device failures within the ECMO space. Alvaro co-leads Dr. Bartlett's lab here at the University of Michigan, and thinks an enormous amount about device functioning as well as creation of devices, and leads an enormity of science in that space over quite a long time. So really, really great his expertise and the team that he's brought around as well to help us think about this. I will make one comment related to this, though, before we hand over, which is just to say that presently some of the data that exists within the EuroELSO registry is being used for device evaluation by regulatory agencies, including the FDA, but other agencies as well. And some of the definitions that are being used there are the existent definitions of EuroELSO, because those are the definitions that are informing whether we identify that a device failure has happened or that a device was exchanged for a given reason. And so we do try, as we have in all cases, to maintain some of that as well. But I think the other piece that I just wanted to highlight is that actually these definitions are being used right now in device evaluation as well. And so without further ado, Alvaro, thank you so much for joining us. Yeah, please, if you have your slides and are willing to share. If not, I'm happy to bring them up as well.
Good morning / afternoon for some of you. Thank you to the organizers, Ryan and Peta, for the invitation. I was able to join. I was worried because I was giving little teaching to high school students here in Detroit how to do hands-on CPR, talking about the basics of ECMO. So now that that commitment is over, I'm happy to go ahead. This is the team that we discussed and came out with the definitions for device failure and adverse events. So I want to start with what is known, and as Ryan was saying, the EuroELSO definitions have most of the components, but not all of the components. And we need to understand that an ECMO system, all components are interconnected into a closed arrangement that ensures blood flow too efficiently into each of them, except for the ECMO console. So we have pump, membrane, lungs, cannulas, tubing, connector, heat exchanger, and our ECMO consoles. The other important concept that is important to understand for this is the failure of one or multiple components can cause sudden loss of blood flow, thrombosis, leaks from blood, plasma, water, can cause air embolism or hemolysis, either of which could cause death, inadequate ECMO support, or damage to any of the patient organs. Finally, a very important issue is that an analysis from the FDA recently about manufactured and user facility devices experienced based on the database, demonstrated that ECMO mechanical failure are associated with serious complications to our patients. The most recent patient-related adverse events that were published are related to hemodynamic decompensation and death, and the most reported failure of components were the pump and membrane oxygenator. With that being said, we are all familiar with the MCSR consensus. They recommended that any definition of device malfunction must capture sufficiently granular data to be meaningful to the clinicians and the engineers. It's important that these definitions can determine the root cause of the malfunction and to develop corrective strategies to identify any problem. As Ryan was mentioning, we use the ECMO definitions that was used for the ECMO registry data definitions. And historically, you all know that ELSO developed failure definitions for most of the components, not all of them. Primarily, it was for pumps, cannulas, oxygenators, and heat exchangers. With that being said, these definitions require a revision. They need to be aligned with the new ECMO paradigm, so the use of ECMO, the proper documentation, the impact of the failure to the patient health. We need to include all the components of the current ECMO system and set out a framework to future clinical trials on ECMO. Finally, ECMO-ELSO definitions then incorporate the ECMO components and the potential degree of failure of each of them. So what we did is we went back and reviewed the ELSO definitions. We reviewed the MCS consensus about the pump, and then we went back and reviewed multiple ISO guidelines, and this is more for device testing and development. So as I said, I would like that you guys consider that the failure of an ECMO system or a component should take into consideration the structure, the material, function, the clinical utility, the usage environment, and the risk benefit. So ECMO Central has moved away from the MSCS RC definitions about device malfunction. And we want to set a new set of definitions that include all the components of the ECMO system and the effect of failure in patient morbid mortality. So these new standardized definitions will facilitate the reporting of adverse events and optimize clinical outcomes, operative proficiency, and existing device technology. So we started with a big Excel sheet. And at the beginning, I told Ryan and Peta and the team that we should go over every single component. Because of time, we started with just 3 components. And when we presented our first components, which were the phase 1, I would say, the ECMO circuit, the pump, the membrane lung, and the cannula. After we presented to the group, everybody agreed that all the components needed to be included. So this phase 2 integrates the ECMO console, the tubing, connectors, and the heat exchanger. And it's very important that we develop a patient endpoint-based severity. And then this is something more that we need to refine, is about the effect, is this iatrogenic versus a patient-related failure. It may or not apply for ECMO in specific components. One of them will be the cannula. And then the other thing that we're working on is to create a survey where you can assess the relatedness of a component failure and its hemolysis versus clotting. So ECMO definition of malfunction, it was organized into 4 worsening degrees or device malfunction. So we have a structural failure, functional failure, device dysfunction, or device replacement without a dysfunction. And we consider the 8 elements of the ECMO system that you see listed here. So the new definitions, this is the summary. Type 1 is a structural failure defined by anything that impacts the integrity of the device separation of blood and air leading to blood leak, air entrainment or embolus as is managed with replacement. Type 2 is a functional failure, basically a mechanical operation. So it's a failure or presumed failure of normal mechanical operation of any component of the ECMO system. Type 3 is a device replacement with evidence of dysfunction. So it's a failure of the device to perform its dedicated tasks at optimal parameters. And type 4 is unique on ECMO, is a device replacement without evidence of dysfunction. So here is the replacement of a device without documented failure of device, more often due to a center-specific protocol regarding longevity of the use of the ECMO system. With that in mind, we also developed the endpoint-based severity. So we have 5 grades. Grade 1, which is transient, asymptomatic, or mild symptoms, clinical or diagnostic observation only. Grade 2 is the maintenance of patient vital signs that require an escalation in non-ECMO life support intervention. Grade 3 is the severe hypotension or hypoxemia in despite of medical interventions that include CPR for more or less than 5 minutes. Grade 4 is a life-threatening consequence with cardiopulmonary arrest for more than 5 minutes. And grade 5 is death or fatality. So for the type 1, we identify the 8 components. So a failure of the pump is replacement due to identify a structural failure of the blood pump, such as the blood leak or entrainment of air. Failure of the membrane lung is replaced due to identify the structure failure in the membrane lung, such as plasma leak, blood leak, suspected rupture or dysfunction of the membrane fibers, resulting in air embolism. A cannula failure was defined as a replacement due to identify the structural failures such as blood leak, entrainment of air, or obstruction to flow, including cannula fracture or indentation. Then we include the tubing, so it's replaced due to identify the structural failures such as blood leak, entrainment of air obstruction to ECMO flow. Tubing connectors is a failure that require a replacement to identify the structural failure such as fracture resulting in blood leak, entrainment of air, or obstruction of flow. Heat exchanger was basically replacement due to identified leak, structural failure that has leaks. The ECMO console was defined as replacement due to identify the structural failures such as hardware or software that leads to cessation of ECMO support. And the whole system, this is primarily designed to integrated systems where we need to replace or impair structural integrity of the integrated system. Now, type 2 is a failure or presumed failure of normal mechanical operation. So the blood pump, we include here the pump head, the motor, or both, replaced to identify mechanical failure of the blood pump. The membrane lung is replaced to identify mechanical failure of the membrane lung that manifests a sudden loss or clinically significant decrease in blood flow. And this is important without modifying the system RPMs, resulting in emergent equipment replacement. The cannula is a replacement to identify mechanical failure of the cannula, manifested as a sudden loss or clinical significant decrease in blood flow without modifying the system RPMs, resulting in emergent equipment replacement. For the tubing, is replaced in response to a sudden loss or clinical significant decrease in blood flow without modifying system RPMs, resulting in emergent equipment replacement. Tubing connectors is a replacement in response to a sudden loss or clinical significant decrease in blood flow without modifying the system RPMs, resulting in emergent equipment replacement. Heat exchanger is a replaced due to complete failure of the device to regulate temperature. The ECMO console is replaced due to an electrical failure or other operational failure of the ECMO console, resulting in replacement due to lost ECMO flow. Example here will be a knob that is not able to dial or a screen that you cannot able to modify. Whole ECMO system, again, is related to integrated system where replaced for impaired mechanical operation, including the ECMO system console in some of them. Type 3, so we have here a failure of the device to perform its dedicated task of optimal parameters. The important key part is here we have clinical evidence of dysfunction. So for the blood pump, we have pump replaced due to hemolysis of a known source. So the pump was replaced due to center-specific markers of hemolysis. And when I keep it open, as you guys saw the previous presentation, there are some centers that cannot measure this, but each one has a way to define hemolysis. B, type 3B will be a pump replaced due to coagulopathy of a known source. So the pump was replaced due to clot burden or coagulation derangement of unspecified source. For the membrane lung, we have in the type 3, 4 subcategories. A is coagulopathy with membrane lung as a known source. So the membrane lung is replaced primarily indicated by clot burden or coagulation derangement within the membrane lung. B is hemolysis within the membrane lung as a known source. So device is replaced indicated by center-specific markers of hemolysis, believed to be related to the membrane lung. C is a decreased efficiency of gas exchange. So the membrane lung replacement for the primary reason of compromised oxygen and or ventilation. and this is typically a more common consequence of a clot burden over time. And D is increasing resistance to blood flow as a membrane loan is replaced in response to increased transmembrane pressure or decreasing blood flow without RPM's adjustment over time. For the cannula, sorry. For the cannula is the visual identification of thrombus within the cannula, and intervention is performed for thrombi removal. For the tubing, it's a tubing connector replaced due to the fibrin or thrombus occlusion. For the tubing connector, it's a connector replaced due to fibrin or thrombus occlusion. Then the heat exchanger is divided also in A and B. Heat exchanger can be replaced due to a loss of expected temperature control. So the heat exchanger was replaced due to a concern that is not maintaining preset temperatures. Or B, heat exchanger can be replaced due to loss expected temperature control. So the heat exchanger was replaced due to causing unintended patient hyperthermia. Then we have the ECMO console. So the console is displayed fails of the ability to adjust ECMO settings via knobs, dials, or touch screen. Alarms are no longer working properly, either the sound or the visual alert. Then the whole system can be replaced, and they have three subcategories, A, B, and C. A, where the entire circuit replaced due to hemolysis of a known source. So the entire circuit was replaced due to center-specific markers of hemolysis. B, the entire circuit is replaced due to coagulopathy of a known source. So the entire circuit was replaced due to a clot burden or coagulation derangement of an unspecified source. C is entire circuit replaced due to indicated components change. So here the entire circuit was replaced as part of a whole circuit exchange, and usually is primarily for an indicated related to the failure of a single circuit component in these integrated systems. Then finally, category or type 4 is a device replacement without evidence of dysfunction. So here is a replacement of the device without documented failure of the device. Most likely is due to a center-specific protocol regarding longevity of the device. So the blood pump, we have equipment longevity center protocol. So the pump is replaced by center-specific protocol regarding longevity of use without evidence of other derangement. This is sometimes seen during the transport of patients or to change the ECLS circuits. The membrane loan, very similar, is equipment longevity center protocol. So the membrane loan is replaced by center-specific protocols regarding longevity of use without evidence of other derangement, maybe due to transition to or from a transport of ECLS circuits. For the cannula, the tubing, the heat exchanger, and the ECMO console, this evidence of dysfunction is not applicable. However, for the tubing connectors, is defined as the replacement of a tubing segment replaced by center-specific protocols regarding or early clot or fibrin burden. And then finally, the whole ECMO system replacement is divided in A and B. It's the equipment longevity center protocol. The entire circuit replaced may be center-specific protocol regarding longevity of time of use without the evidence of other derangement. Again, possible due to the decision to or from a transport ECLS circuit. And the last one is type 4B, where the entire circuit replaced following temporary transition to bypass. The entire circuit is replaced during the whole circuit exchange following a temporary transition of patient mechanical support to CPV with a continuous ECLS run. So with that being said, those are the definitions that we came out with.
Thanks, Alvaro. I think it's really easy to get into the weeds of this because we've spent a lot of time looking at these definitions. But one of the things that I just wanted to highlight was the hierarchical nature of the different failures that you described. So the structural failure, then the functional failure, and then dysfunction of device, and then swapping out devices without dysfunction is kind of the key elements that we wanted to bring through. But thank you so much for going through that. And I'd love to hear questions and comments. And in the meantime, I think Ryan's going to pop up a QR code that we're hoping that anybody who would like to make comments on any of the definitions you've seen or the process, or that you'd like to receive the definitions for comments, please get back to us on the SurveyMonkey that will be linked to this QR code.
Also, as we're waiting on any questions as well, I think the other thing I would just mention is that in Alvaro, you said this, but I think, I've recognized that we've said lots of things and sometimes they're not always heard because we're saying so much. So there's the part that Peta just reemphasized in terms of the scaling of the structural failure. And then side by side of that is what the impact of that device exchange is to the patient as well. And that's that other slide that Alvaro was showing, which is around like, hey, did it have just some minor physiologic down to, did it cause CPR down to death as well? And so those things as well.
Barbaro, one thing that for me was really catching is the fact to classify the type 4 changing a device when you don't have a real failure. Some more comment on this?
Yeah, like why we included that in the device malfunction. I think that's a really good point. So I think,
yes, agreed.
Yeah, so I think, clearly there are times where, between the kind of, there's the structural failure where like there's something's broken and it has to be changed. There's a sudden shutdown of the system because of a mechanical failure and it's like you have to change stuff. And then there's dysfunction, where you have things that are wrong or you're worried about and you want to change. And then I think there are centers that have protocols where they're changing either based on manufacturer indications for length of use or based on, center specific recognition of how long they feel comfortable with. And I think the reason we ultimately have decided to keep this is, and the FDA was pushing back at us about this. And it's like, if you're changing it, you probably think something's wrong. I'm not sure that's always true, but I think it's possible that's true, that you're saying, I don't actually think there's a dysfunction, but maybe there is some element of softer dysfunction that you're less clear about. But the other thing is the second part that we talked about is what happens to the patient. So it's possible that you've decided to change this device because you have a protocol that indicates you should, and then the patient suffers a rather significant consequence, such as a cardiopulmonary arrest. And we wanted to have the opportunity to capture that if devices are being switched out, regardless of the reason, that a patient impact could be experienced. And we thought that was important to capture as well. Alvaro, do you have other comments there?
Matteo, that was a great question. And I think Ryan answered it. The second part was the kind of the strength. So we can gather the data, specifically if you're going to use this for clinical trials, because there is a lot of papers now saying that the major complication on ECMO is when you do your circuit exchanges. So if we can at least gather the data and say, okay, maybe there is no need to have this indicated, but we need to capture it. I know it's not the best, but the need is to capture the data.
Yeah, Matteo, sorry, I think you did say Alvaro. I heard Barbaro because I'm just recognizing now our names end in the same sound and I just caught the back bit of it. So sorry, Alvaro.
All good.
My apologies.
Yeah, 2 Italian names.
I can see that somebody in the attendees list has their hand up, but we've got no way of calling on you to speak. And so if you could type your message into the chat, into the Q&A, we'll be able to answer your question or respond to your comment.
Sorry. While that's happening, I think the other thing I just want to express is just a huge gratitude for everyone joining today and helping us think about this. And also everyone who's been a part of this, as Peta mentioned, in addition to the leads that aren't presenting today, there are teams within those leads. There was so many folks who have responded to us to just talk through things with us and help us think about things. Too many for us to start to walk through those names, but we have just been absolutely inspired and incredibly grateful for the willingness for people to help us think about this. I do think it comes back to what Alain and Matteo were saying at the beginning, that this is really important work for us to be able to help to organize our clinical research going forward, but it needs to be all of our work. And so thank you so much for the willingness to help us make that a reality.
I think we have a couple of minutes before closing. I don't see other question. Do you have some text in your laptop?
No, I've got no additional text.
So let me congratulate with all the speakers. What you listened today was just a part of a brilliant work that this colleague have done in those months. I'm very happy to have spread in Europe today this great initiative. I hope that in the future it will help to harmonize definitions and to provide better statements, not only for the research, but also for us working at bedside to understand each other more clearly. Thanks, Ryan. Thanks, Peta. Thanks all the speakers in line. Thanks, Aparna. Thanks, Peta, Alain, Peter Shelongowski to have been present online. And thanks to EuroELSO for hosting this webinar.
Yes.
Thank you so much for the opportunity to talk to you all today and for joining. We really, really appreciate all the feedback and look forward to more through the SurveyMonkey.
Yes, thank you, thank you, thank you.
Thanks all, guys. Thank you all. Have a nice evening or journey.
Thank you.